Synthetic technologies allow scientists to venture into uncharted waters, asking unique research questions and finding previously unattainable solutions to some of life's biggest mysteries. From gene editing to protein engineering, synthesized DNA libraries enable researchers to grasp once unreachable high-throughput screening applications and dismantle barriers between experimental ideation and execution. In this podcast series, Synthetic Screens, Genes, and De Novo Proteins, the Scientist Creative Services team talks to experts about their experiences implementing Twist Bioscience's synthesized long double-stranded gene pools, called Multiplexed Gene Fragments, for high-throughput screening. In this episode, Deanna MacNeil from The Scientist spoke with Josh Tycko, a neurobiology postdoctoral researcher in Michael Greenberg's laboratory at Harvard Medical School, about investigating gene regulation with synthetic DNA libraries.
Cells finely tune gene expression to meet their unique fitness needs in a given tissue or cell culture context. In the current era of genomics research, scientists have made impressive strides toward figuring out where different transcription factors bind DNA to regulate gene expression. Mapping out the human genome across several different cell types to identify locations of active promoters and enhancers, and denoting their respective transcription factor partners. But beyond where transcription factors are going, how these proteins do what they do remains unsolved. Building on his professional and academic background in synthetic biology and gene editing, Josh Tycko investigates effector domains, which he calls the business end of transcription factors. These protein domains have less of an effect on where a transcription factor goes and instead determines whether it functions as an activating protein, a repressor protein, or in some cases, both. Tycko takes a systematic approach to mapping transcriptional effector domains across different proteins with the help of synthetic DNA libraries that encode thousands of effector domains. And he created a high-throughput method that bridges the strengths of existing protein engineering techniques.
Transcriptional effectors have been studied for a while, especially in a one-at-a-time individual way with what's called a recruitment assay. This is really an old school synthetic biology approach. You're engineering a new protein. It has a standardized DNA binding domain that is known to bind a certain location in the genome. Onto that DNA binding domain, you can fuse a new transcriptional effector, and it will be recruited to that position in the genome. You can measure gene expression from that target and see, did the transcriptional effector increase or decrease expression relative to a control. We realized we could bring this recruitment assay into the high throughput pooled screening era. And to do this, we leaned on some approaches that use DNA libraries synthesized at a large scale. We could make a library that encodes thousands of different candidate transcriptional effector domains and then recruit those domains to a target gene in a way where we are testing one candidate effector domain in each cell, but we're growing millions or hundreds of millions of cells.
This method, called HT-Recruit for high-throughput recruitment, allowed the researchers to separate cells based on candidate effector-driven gene expression changes. After collecting cells with high or low target gene expression, Tycko generated sequencing-based scores for each effector domain in the large-scale library, quantitatively measuring the transcriptional effector activity of thousands of human protein domains and mutated variants. Tycko recently paired HT-Recruit with CRISPR-I-based DNA binding domains, such as CRAB domains, which allow scientists to recruit proteins encoded by transcriptional effector libraries to endogenous sites in the genome and investigate context-dependent gene regulation in a high-throughput manner. These new approaches to studying gene regulation are made possible by advancements in synthetic DNA library technologies.
The most enabling research tool for HT-Recruit was the new development of oligonucleotide libraries that contain longer pieces of DNA. We needed to use oligonucleotides that were 300 base pairs long so that they were long enough to actually encode full transcriptional effector domains. Until that project, the only oligonucleotides libraries that I had seen were quite a bit shorter. Those short oligos were really useful for screens to study the necessity of different genes for a phenotype. When these longer pieces of DNA became available and it became possible to encode really full-length domains, that was very exciting because now you can do this different type of sufficiency screen. If we overexpress a domain and recruit it to a target gene, is that sufficient to activate or repress a target gene? Just hearing that these 300 base pair pieces of DNA were becoming available is honestly what inspired us. Like, now is the right time to do this HT Recruit project.
With 300 base pair oligonucleotides, Tycko is able to encode an 80 amino acid domain with wiggle room for cloning adapters on either side. Although this falls short of a full-length protein, 80 amino acids are enough to encode roughly 70% of annotated domains present in human nuclear proteins, including domains with well-characterized behaviors, such as the crab domains that the researchers use in their experiments. For the other 30% of protein domains, researchers need tools that span longer DNA stretches to fully investigate their roles in gene regulation. There are annotated protein domains that reach over hundreds of amino acids, which scientists may be able to subdivide into smaller, easier-to-study pieces with a bit more characterization. However, these longer domains have historically been out of reach for pooled screening methods like HT Recruit. Researchers can turn to Twist Multiplexed Gene Fragments to overcome this length limitation.
More recently, we've been able to start using these multiplexed gene fragments, MGFs, which, taking this idea of these long oligonucleotides even further, going out to 500 base pairs, that ability to make longer DNA libraries inspired us to be creative. Like, what are new projects that are possible with this new capability? For one, it's really exciting because we were missing 30% of annotated domains at the previous length limits. Another new possibility that's been exciting for me is combinatoric. In full-length proteins, there's generally more than one domain, and these work together in some way to achieve the protein function. In the past, before the MGFs, what I needed to do was try to come up with ways to clone libraries where we could make one domain at a time, fuse them together with some type of linker, and study their function. We could find how different domains synergize together or buffer one another's functions using those types of combinatorial libraries, but it's definitely a more difficult undertaking. And the ability to just program, like type out in the computer, the combinations of domains that are interesting to study and then just have them printed out in one go, it makes that process a lot easier. The other thing is that longer DNA library synthesis is important for the non-coding genome space as well. Thinking about regulatory elements like enhancers and promoters, these have been studied quite deeply at the scale of 120 base pair libraries, which is interesting for telling you what like a few kind of transcription factor motifs together can do. But in reality, many of the regulatory elements in the endogenous genome or that are used in gene therapy and synthetic biology research are much longer than that, often 500 bases or more. And I think that's because they really rely on the combinatorial function of multiple transcription factor binding sites within that stretch of DNA sequence. So we've been excited about these longer fragments with MGFs, allowing us to encode closer to the full regulatory element. And I think this is especially exciting for finding regulatory elements that are more finely tuned to drive gene expression in a quite specific cell type.
In ongoing research, Tycko is applying these libraries in vivo in the brain, where the presence of many different cell types poses a pressing problem for finding regulatory elements with finely tailored expression in just one or a few of the cell types. Because multiplexed gene fragment sequences are initially synthesized as user-defined oligonucleotides and then amplified as long double-stranded DNA fragments, they make it easier for researchers to recapitulate more of the endogenous sequence and make certain experiments more likely to mirror how a sequence would behave in its endogenous setting. Tycko is also interested in using these DNA libraries to take an unbiased approach when identifying transcriptional effectors and other types of protein domains. Instead of domain-focused library design, which begins with existing annotations of where domains occur within proteins, unbiased library design characterizes functional domains by chopping proteins into tiled fragments, irrespective of known domain locations. Pools of synthesized tiles that encompass entire coding sequences allow scientists to discover functional domains in otherwise unannotated regions. But these experiments can be technically challenging and costly, depending on the tile length and total region of interest.
These end up being really large libraries. You need to make lots and lots of tiles in order to cover the full coding sequence of the proteins that you're studying. For example, using over 100,000 tiles, we were able to completely cover the known transcription factor proteins and thus feel that we were systematically finding all transcriptional effector domains within those proteins that fit within this tile size and work in the context we tested. But that was a difficult experiment to do. I think if you wanted to make a tiling library that covered all of the human proteins to really systematically find functional protein domains within the full proteome, you're talking about like over 1 million tiles, which is exciting, and that's not totally outside the realm of possibility, but definitely a difficult and expensive thing to do. That problem does get easier as these tiles get longer. So if your tiles can be longer and you can have a wider kind of step size between tiles, you can comprehensively cover the full space of protein sequences without having to synthesize and clone and measure quite so many individual tile elements, which could make some of these ideas more feasible.
Multiplexed gene fragments allow scientists to ask and answer bolder scientific questions, bringing high-throughput techniques not only into the realm of possibility, but into the realm of successful discovery and beyond. Tycko looks toward the future possibilities in synthetic biology and gene regulation research enabled by longer pooled DNA libraries that nearly stretch the imagination. Once available tools make ideas possible, the sky is the limit for experimental design.
This is an awesome space for expressing creativity in science. The number of questions you can ask using synthetic libraries is probably boundless. You can just try to think about your favorite protein family and how you might design a new reporter system to study their protein functions. And using these synthetic libraries, a single person in a single experiment can really map function across an entire protein family in a relatively feasible way. It just opens up so much space for not only pursuing a hypothesis you might have about your favorite protein within that family, but for also discovering the unexpected. For example, the crab family of domains, these are known to be repressors. They're used in the CRISPR-I system for silencing genes. But when we used a synthetic library to study all of the crab domains, we found that actually a few of them are activator domains, doing the total opposite in some sense. One of them was actually the strongest activator domain we found out of any activator. And that unexpected discovery of a really strong activator within the crab family was interesting for better thinking about how repressors and activators evolve. Also, we used it to develop a new synthetic biology tool. That was just a very cool path that I think we wouldn't have gone down without having the synthetic library that enabled us to cover that whole space. I think there's a lot of exciting room now to work on improving the techniques that we use to deploy synthetic libraries. For one, are there ways we can get around using these dangerous and high biosafety level viruses to install libraries into cells? With these new, longer multiplex gene fragment libraries, one thing I've been playing with a bit is like maybe we can use recombinases to just directly install these double-stranded DNA libraries right into a safe harbor site for expression in a cell line without having to make lentivirus and do large-scale cloning. I think there's lots of room for new techniques on getting these synthetic libraries into cells in vivo and coupling their functional readouts with the identity of the cell type that they're delivered to. So whether it's on the questions you're asking or it's in further improving the tools for using these synthetic libraries, I just think there's a lot of places for people to be creative in this space.
Thank you for listening to Synthetic Screens, Genes, and Novo Proteins, brought to you by Twist Bioscience. And thanks again to Josh Tycko, postdoctoral researcher at Harvard Medical School. Join us for our next episode where we discuss de novo protein design with Jeffrey Chang from Harvard University. To keep up to date with this podcast, follow The Scientist on Facebook and Twitter, and subscribe to The Scientist Lab Talk wherever you get your podcasts.